So my name is Jørgen Ankill and I'm a PhD student at the Department of Cancer Genetics at the Norwegian Radium Hospital in Oslo. So in the poster presented, we aim to get a better understanding of the role of alterations in DNA methylation patterns on breast cancer pathogenesis. So the hope was that we could identify previously undescribed transcriptional networks that are dysregulated by alterations in DNA methylation patterns and identify key cancer-promoting epigenetic alterations that might represent lucrative targets for epigenetic treatment in the future. So previous studies have shown that alterations in DNA methylation occurs early during breast cancer carcinogenesis. So in brief, DNA methylation is an epigenetic modification that mainly occurs at CpG sites in the genome and is well known to regulate gene expression when found in regulatory regions. So for instance, DNA methylation at promoters is usually associated with gene repression, while the opposite is true for unmethylated promoters. In addition to promoters, DNA methylation at intergenic regions such as enhancers have also been shown to be associated with gene expression. So these are regulatory regions in the genome that may be located far away from their associated gene, but may interact with the gene promoters through transcription factor binding and this formation of this chromatin interaction loops. But still little is known about the role of DNA methylation at enhancer regions regarding transcription factor binding and breast cancer pathogenesis. So in order to identify transcriptional networks that are dysregulated in breast cancers due to alterations in DNA methylation patterns, we performed genome-wide expression methylation quantitative trait loci analysis, or emQTL analysis, which is a method that had previously been published by our group. And this is the outline of the emQTL analysis workflow. So what we did in brief was to correlate the DNA methylation values for more than 180,000 CpGs with expression level of about 18,000 protein coding genes. And from this, we were able to discover around 16 million significant CpG gene associations in the Oslo 2 breast cancer cohort after bone Ferroni correction. And after validation of these associations in the TCGA breast cancer cohort and filtering of the emQTLs based on a number of associations, we're then left with around 10 million associations, including almost 5,000 genes and 44,000 CpGs. We then grouped the emQTL CpGs and genes by spectral co-clustering of the correlation coefficients based on negative associations, and we discovered 5 biclusters as shown in this heat map. So the biological relevance of the biclusters were then assessed by gene set enrichment analysis. So first of all, we found that we were able to recapitulate the immune and the estrogen biclusters, which have been described in the previous paper. But we also identified 2 novel biclusters, one which was related to EMT, but our analysis has shown that the bicluster is mainly caused by varying degree of fibroblast infiltration, and the other one was associated with cell cycle related functions, and we'll mainly focus on this bicluster for the rest of the presentation. So we next assessed the DNA methylation level of the cell cycle related CpGs, and we found them to be less methylated in the basal-like and the HER2-enriched breast cancer subtypes compared to the luminal ACE in Oslo 2. And interestingly, these subtypes also had a significantly higher expression of the genes in the cell cycle bicluster. And this is very in accordance with the current knowledge, as these subtypes with the highest expression of these cell cycle related genes are also well known to be the most proliferative breast cancer subtypes. And a similar pattern was observed in the independent TCGA breast cancer cohort as well, both for the methylation and for the expression. So to assess the functional relevance of the CpGs in the cell cycle bicluster, we investigated whether they were enriched in particular regions of the genome or not using chrome-HMM segmentation. And from this we found that the CpGs were significantly enriched in active intergenic enhancer regions of all breast cancer subtypes. And since enhancers are well known to carry sequences recognized by transcription factors, we next investigated whether the CpGs were enriched in the binding regions of specific transcription factors by using Unibind, which is a database of direct transcription factor DNA interactions. And we found the CpGs to be enriched in the binding region of several transcription factors such as ZPB, FOSL1 and FOSL2, which all have previously been linked to proliferation in breast cancer. So since the CpGs were enriched in enhancer regions, we also assessed whether the emQTLs in the cell cycle bicluster were enriched in chromatin loops using experimentally defined GFET pool 2 and predicted EMPET chromatin interaction data from an ER-positive and ER-negative breast cancer cell lines respectively, and we found a significant enrichment in both of them. We then assessed the link between DNA methylation at the CpGs and the expression of genes in the cell cycle bicluster. So we correlated the average DNA methylation level of the cell cycle bicluster CpGs with the average expression of the genes, and we found a significant negative association both in ER-positive and ER-negative breast cancers, suggesting that the regulatory mechanism that we discover is independent of ER state. So by knowing that enhancer methylation at regions of transcription factor binding is associated with the regulation of genes associated with proliferation, and knowing which transcription factors that were involved, we could refine the emQTL approach to more efficiently identify key drivers of proliferation, which could be of potential interest as targets for epigenetic treatment. The refined approach has several filtering steps in order to identify such candidates. So first of all, the emQTL CpG had to be in opposite side of chromatin interaction loops and must be in an enhancer region according to ChromaHMM in one of the subtypes. The CpG had to be in the binding region of any of the rich transcription factors, and the gene must be a part of a curated gene set associated with proliferation. So altogether we identified 53 such candidate emQTLs. And this table basically shows one of the candidates with the strongest negative correlation between DNA methylation at the candidate CpG and the expression of the associated gene in OSTLO2. And the emQTL CpG is found in the binding region of several members of the FOS family of transcription factors, in an enhancer region of several of the breast cancer subtypes. And this region have also been shown to interact with the promoter region of PIM1, which is a gene associated with proliferation. And this figure here on the right hand side shows the correlation between the expression of PIM1 and DNA methylation at the CpG found in the enhancer region. And we can clearly see a very strong and negative correlation both in ER-positive and ER-negative tumors independently. We report genome-wide loss of enhancer methylation at transcription factor binding regions of proliferation-related transcription factors with contaminant high expression of proliferation-related genes that are wired by chromatin looping in aggressive breast tumors. We also identify strong proliferation-promoting epigenetic alterations that represent lucrative targets for epigenetic treatment in the future, and this is something we want to assess further by performing targeted DNA methylation at these sites using CRISPR to assess its effect on proliferation in cell lines. A preprint of the manuscript is now available from bioRxiv for those of you who want to read a bit more detail about the work.